Using sequencing and other tools, we and others have been able to sequence the weird microorganisms inside the guts of sheep or goats or cows and learn that there's really powerful enzymes that can break down cellulose, hemicellulose, and maybe even lignin inside of those systems. My name is Michelle O'Malley, and I'm a professor in the Department of Chemical Engineering and also in the Department of Biological Engineering at the University of California, Santa Barbara. And it's been my pleasure to collaborate with JGI in a number of CSP and ficus projects throughout the years.
Hi, I'm Tejas, Navaratna. I'm a postdoc in UC Santa Barbara, working with Michelle O'Malley with anaerobic fungi. So biomass, plant matter, it's evolved over hundreds of millions of years to be tough, woody, hard to break down. It's polymers, cellulose, lignin, they're pretty complex. Commercially, what's really done most of the time is subjecting this biomass to high temperatures, high pressures, using catalysts, things that are expensive, things that require a lot of energy input, and things that ultimately aren't necessarily as sustainable as using what nature has already devised in terms of organisms that already have enzymes that can break these things down.
Gut microbiomes, especially from herbivores, are a great source for enzymes to use for breaking down plant biomass, because if you think of, for example, a sheep or a goat, they're going to eat everything, and so their digestive system is really primed to get and extract energy out of things that we always think of as trash. We got involved with JGI really early on, almost a decade ago when I started my lab at UCSB. We sequenced some of the first genomes from anaerobic fungi that are resident to large herbivores. And we used that sequencing to really give us clues about what enzymes were there, and that led us down a path of trying to characterize the function of these enzymes and partner with other people at JGI and also at the Environmental Molecular Sciences Lab, or EMSL, to look at what these enzymes are doing, how they're doing it, and trying to engineer enzymes and biosynthetic pathways. And all of that has led to an explosion of collaborations.
What I'm interested in doing is developing these genetic tools, so using CRISPR, using Cas proteins to find interesting sites in the genome and put in other genes, or knock out certain genes that we don't need them to have in order to better and more efficiently produce things we want them to produce, so biofuels and other things.
So a lot of the initial enzyme discovery work that we did from anaerobic fungi that have come from zoo animals and other grazing animals has really led into a better understanding of the whole process for how these microbes do their work, both from how they take in sugar, so studying transporters, to looking at the biosynthetic pathways that help make not only the enzymes but natural products that are key to how these fungi function and might even be a recipe for making new antibiotics to regulate community structure. And so it's really gone off in so many exciting new directions, and it wouldn't have been possible without JGI.